Hello, everybody. We are going to continue with our um, notes for Unit 8. These are the meiosis notes, number 3 in our series. So let's get started. You'll notice this is a Wiser Me worksheet. So when I tell you to pause the video, go ahead and do that and scroll down to answer the relevant question. So the objectives that we're covering in this video are number 5 and number 6. So number 5 tells you to draw a basic diagram of the two divisions of meiosis. So these are called meiosis 1 and meiosis 2. Finally, something that's simple, right? And then to explain how the chromosome number is reduced by half. When you are perhaps filling out your objective expansion for this one, be sure to talk about the shapes of the chromosomes. That will help. Explaining how they go from 46 chromosomes to 23. You'll have to mention whether it's an X-shaped chromosome or an I-shaped chromosome. So keep that in mind as we go through these notes. For number 6, it says describe crossing over that occurs during meiosis and it's important to evolution. So this one is going to tie back to the whole idea of genetic variation. So pay attention to that as we go through the notes. The first one is kind of your brain warm-up. This is a discussion question on the Wiser Me worksheet. So how is mitosis or meiosis different from mitosis? Go ahead and pause the video and put in a comment below. You can agree with someone's comment or create a new one, or if someone, there's only so many things. So if someone's already said something, you can repeat. Go ahead and pause the video and do that now. Great. So hopefully you saw or entered some good things that are the differences between meiosis and mitosis. So let's discuss those now. This graphic is really good. So if you want to pause the video and actually do a screenshot of it, or these videos are all posted on my YouTube channel. There's a special playlist that is the Unit 8 notes. So you can pull it up there later also. So meiosis and mitosis are different. This is a good summary of the ways that they are different. So in mitosis, we started with a normal body cell, a somatic cell, and it essentially replicates itself. So the daughter cells are identical to the parent cell, and the daughter cells are identical to each other. They are also diploid cells. And this takes place over the course of your entire life and of any organism. So from the time that it's a little bitty fertilized zygote, mitosis is what causes the organism to grow and get bigger. This is how an organism increases in size. It's also how an organism replaces cells that are damaged or worn out. Mitosis is a type of asexual reproduction. You do not need the involvement of another cell. It really just sort of clones itself. So flipping over to meiosis, how that's different is that the end result, well, first of all, it goes through 2 divisions instead of just 1. So this is just one division. Meiosis goes through 2 divisions. The first division right here between the germ cell and the first 2 here, this is meiosis 1. And in meiosis 1 here, we have all of the phases. So we have the 4 phases, prophase, metaphase, anaphase, and telophase, and cytokinesis. So all of those are called meiosis 1. And we actually reference them as like prophase 1, metaphase 1, anaphase 1, telophase, cytokinesis 1. And then after that occurs, there is another division that reduces the chromosome number again. This second division is called meiosis 2. And again, it goes through prophase, metaphase, anaphase, and then telophase and cytokinesis. And we indicate those with a Roman numeral 2 after it. So prophase 2, metaphase 2, anaphase 2. You're not going to have to identify or label those phases like we did for mitosis, because in fact they're very similar. There are some key differences. You'll notice that at the end of the first division in meiosis, you still have the X-shaped chromosomes. So what happens here, instead of pulling apart the sister chromatids, you'll see when we talk about crossing over, in prophase 1, the homologous pairs, so the X-shaped mom chromosome and the X-shaped dad chromosome actually line up next to each other, and then those are separated. So that ensures that we get an X-shaped number 1 chromosome and an X-shaped number 2 chromosome and an X-shaped number 3 chromosome all in each cell. Some of them are going to be from mom and some of them are going to be from dad. And you'll see when we talk about crossing over, a lot of them will be a mix of both. So in this first division, we separate the homologous pairs. And that's something to note. So write down that in the first division, we separate the homologous pairs. Say it one more time. In the first division, we separate the homologous pairs. In the second division, we then take those X-shaped chromosomes and we separate the sister chromatids like we did in mitosis. So then we end up with 23 chromosomes, I-shaped chromosomes, in each of the resulting cells. And I'm going to post some good review videos on this if you want to go through and take a look at and understand exactly what's going on in each division, so meiosis 1 and meiosis 2. I'm not going to get very detailed in that. What you need to know, again, is in the first division, we separate the homologous pairs, and in the division 2, we separate the sister chromatids. So that's about as much detail as you need, but I will post some videos that go into more depth if you're interested or you feel like you need that background in order to understand what's going on. So your objective is to draw a simple diagram that shows what's going on with the chromosomes. So here is a really good example of that diagram. You'll see we have meiosis over here on the left, and we start out with, this is called a germ cell, and it has 46 X-shaped chromosomes. Just like mitosis, it has 46 X-shaped chromosomes. Remember that these X's are duplicated chromosomes that happened in S phase of the cell cycle. So that's a throwback to the last unit, but the left side of this X is an identical copy of the right side of this X, and it was formed during S phase in the cell cycle. So now it is entering meiosis, just like it was entering mitosis after the critical checkpoints in G2, where the chromosomes are proofread. All right, so they start out the same, 46 X-shaped chromosomes. In the first division of meiosis, we line up all of the homologous pairs. So we'll have an X-shaped chromosome that represents number 1 from dad and an X-shaped chromosome that represents number 1 from mom. These two line up together, and then in the first division, those X's are separated. So now we have 23 X's in each cell. If you remember, every cell in your body has 2 copies, right? We have 23 pairs. And that's what we mean when the homologous pairs are separated. So we end up with 23 in each cell, and it represents 1 pair of 1 out of that pair. So there are still copies here though, right? if this is the number 5 chromosome, it still has an identical copy. So now in meiosis 2, we're going to pull that X apart so that we end up with 23 I-shaped chromosomes for this cell, and we end up with 23 I-shaped chromosomes in 2 cells from this one. And this is how we go from 46 chromosomes down to 23. We decrease the number of chromosomes by half, because in mitosis, when we just make a copy of the cell, you have your 46 X-shaped chromosomes, you pull them in half, and now you have 46 I-shaped chromosomes, which are identical to the parent. These are the cells that would then go into G1, where they increase in size and they just sort of grow and they do their thing, and they go into G0, where they carry out all the normal functions of life with the proper number of chromosomes, which is 46 I-shaped chromosomes. When they're signaled to divide again, they go into S phase, where you end up going from a 46 I-shaped chromosome to a 46 X-shaped chromosome in the S phase. Meiosis is different because now we have, in the final product, we don't have an exact duplicate of the original cell, which was 46 I-shaped chromosomes before it went through S phase. So this is an identical copy of the parent cell. This is not. This is 23 chromosomes. Our cells need 46 chromosomes in order to function properly. And that's where the fertilization comes in. And that is why meiosis is considered sexual reproduction. The diagram that I have here is showing spermatogenesis, which is how sperm cells are created. It works a little differently for female egg cells, which I'll get to in a little bit. But so these would be 4 sperm cells, and it has 23 chromosomes in it. So when it merges with an egg cell, which also has 23, we end up with the correct number of chromosomes being 46. So this is the diagram that I'm talking about in the objectives. So there's something special that happens during prophase 1. When those homologous chromosomes line up together and before those pairs are separated, they actually end up mixing up their genes. And this is one of the really big evolutionary advantages of sexual reproduction during meiosis, and it happens before the sperm cells and the egg cells ever get together. So what happens here in prophase 1, we have the X-shaped chromosomes, and these are homologous. So we're going to say that one's from dad and one's from mom. And these chromosomes, one from mom and one from dad, they line up together. Now, remember, the definition of homologous means they have the same type of information on them. So if we pretend like at the very top here, the gene for eye color occurs at the top part of the chromosome, then the trait for eye color would occur at the top part of this chromosome as well. So they are the same type of chromosome. And they pair up, and then they do something really cool. They actually swap DNA. So here's a video to kind of animate this. So this is meiosis 1, and then here's meiosis 2. So you end up with 4 sperm cells. Some of them got the mom chromosome, some of them got the dad chromosome. Here's crossing over. So what you see here is, I like this animation, and you can rewind it and play it again, but it showed the two divisions. In the first division, the purple chromosomes stayed together as an X shape, and that was meiosis one. In the second division, those two purple chromosomes separated, and that's meiosis two. What's unique about crossing over and how it increases genetic variation is that this chromosome right here is essentially intact. It's the same chromosome that you may have, or that if this is the mom version, that your mom has. However, this chromosome is a unique mixture of your mom and your dad, which is how your children can have traits of both your mom and your dad at the same time. This chromosome right here, this particular collection of alleles, has never existed before. Take a moment to think about that, because your mom's chromosomes and your dad's chromosomes swap some information. The resulting chromosome is completely new. It's part of the reason why no two siblings have exactly the same DNA. The DNA gets mixed up and shuffled at the chromosome level before or during the creation of the sperm cells and the egg cells. And what's even cooler is that where they cross over and how they cross over is completely random, and it's completely different every time. So no two sperm cells, so if these are four sperm cells, no two sperm cells are ever exactly the same. Now this example is using just one chromosome. How many pairs do you have? 23. So that means that crossing over occurred with 23 different chromosomes, all getting a completely random and unique mix-up of genes from your mom and your dad. It's kind of crazy, huh? So your egg cells, none of them are the same. None of them. So even if every one of your egg cells became children, none of them would be identical in terms of DNA, which is part of the reason why using DNA to identify someone is a very powerful tool. In all of human history, those chromosomes have never existed before. It's kind of cool to think about, because it's happening, and the animation shows that it's happening on just the lower portion, but it can actually happen here and here. You can also have it where it just takes a chunk out of the middle. So you can have a blue piece in between two purple pieces, and it's completely random every time. So pretty cool. All right, we are going to move on. Here is a sort of a picture version, and I like this 2D sort of animation because it shows the homologous chromosomes, so the mom copy and the dad copy line up together. Again, this is in prophase 1. And then they wrap their little sister chromatids around each other, and there are bits of chromosomes that are swapped. So the genetic information is actually flipped from the mom chromosome to the dad chromosome in order to create new variations. Now, in terms of evolution, these alleles are not unique, right? Like your mom had that allele or your dad had that allele. However, the combination of alleles is new. And that new combination of alleles could result in new or different traits, because there are very few traits that are just one allele. In fact, like your eye color is 15 different genes. So out of those 15 different genes, maybe 3 of them are from your mom, 4 of them are from your dad, and the rest are some sort of mix, right? So genetics gets extremely complex. especially when we start looking at the DNA and the different combinations of alleles. So different combinations of alleles can result in new or different traits as well. It doesn't necessarily have to be a mutation, like a new allele, but having different combinations can create a new trait as well, especially when you think about the fact that most traits rely on more than one gene. So if you can mix up those different combinations of those 15 genes that create eye color, even though the individual information isn't new, you've got new combinations of that information. So how does this work with humans? Um, and essentially anything that creates an egg and sperm. So the sperm generation is called spermatogenesis, genesis meaning the beginning, so the beginning of sperm. And the top picture up here talks about how you have the germ cell, the original starting. You go through meiosis 1, where you separate the homologous chromosomes. You go through meiosis 2, where you separate the sister chromatids, and you have 4 mature sperm cells. It happens a little bit differently in egg cells. In egg cells, you end up with one egg cell out of the whole process, but it's important to go through the entire process because you have to reduce the chromosome number with 2 divisions, and it also allows an opportunity for crossing over to happen in meiosis 1. So why does it only produce one? The answer lies in the size and the eventual function of the egg cell. When the sperm and the egg cell meet and fertilize and create a that zygote, that potential offspring, must have all of the cellular components to start going through growing, dividing, and going through mitosis. That means it has to have centrioles, it has to have spindle fibers, it has to have Golgi apparatus, it has to have endoplasmic reticulum, it has to have all of the organelles necessary for that fertilized zygote to grow into an embryo. So what happens here, if you look at the diagram, we start out with the same germ cell, and in the first division, we line up the homologous pairs, so the mom and dad copies, and we do separate those. But when we separate those, all of the organelles and most of the cytoplasm and all of that stuff goes to one side. So when it splits, it's not equal. So you would think like when it splits, you get 2 of equal size, and that's not the case. When it splits, you get like a really big one and a really tiny one. And the really big one has all of the organelles and all of the stuff that it needs to grow into an embryo. Well, that's the first division. The same disproportionate division happens in meiosis 2 as well. And all of the organelles and all of the stuff that the cell needs in order to create an embryo go into one cell or the other. The little one is called a polar body. So the little ones they're called polar bodies, and they only contain DNA and a little bit of cytoplasm. And those are actually broken down and reabsorbed into the body by the immune system. They're broken down, all the pieces and parts are reused. And then it's that one egg cell that contains all of the necessary material to create an embryo. For sperm cells, they really just contain DNA and some cytoplasm. There's no organelles involved. Which is why when we talked about the mitochondria, all of the mitochondria you have in your body came from your mother, because all of those mitochondria were pushed into that one egg cell. There are no mitochondria in the sperm cells. It's not. The only thing in there is DNA. And that's it. So when we create sperm cells, there are 4. When we create an egg cell, there's only 1, because that egg cell needs to have all of the organelles and all of the stuff necessary to create an embryo. So I have another discussion question. So pause the video and scroll down to the next discussion question, which what might happen if the chromosomes don't separate correctly or are damaged during meiosis? So pause and go answer that. All right, welcome back. So what can happen? Here's meiosis 1, where those homologous chromosomes line up and then they get separated, right? So here it's showing kind of anaphase where those homologous chromosomes are being pulled apart. And then in meiosis 2, we pull apart those X-shaped chromosomes and create I-shaped chromosomes. Well, what if one of the X-shaped chromosomes doesn't separate properly and both sides of the X go to one cell versus the other? So on the right is how it's supposed to happen. Those X's get separated and we end up with, two different chromosomes here. On the left, we have the smaller chromosome separates correctly. So we have a small chromosome in each of the cells. But the larger chromosome wasn't pulled apart properly. This means that you end up with an egg or a sperm cell that has an extra chromosome. So if this were the number 2 chromosome, this egg, when it gets fertilized, it'll have 2 number 2 chromosomes from mom. And then when the sperm comes along, it'll end up with 3 total because the sperm will add another number 2 chromosome. And so there will end up being 3 number 2 chromosomes. This egg is missing that number 2 chromosome altogether. So this egg cell is missing the number 2 chromosome. When the sperm cell comes along, it only has 1 number 2 chromosome. So the baby that's produced will be missing a chromosome. It'll be missing the number 2 chromosome from mom because that ended up in a different cell. So what does this mean for humans in particular? Well, this can result in a lot of chromosomal disorders. That's what they're called, chromosomal disorders. And hang on, my nose itches. These chromosomal disorders, sometimes they result in a miscarriage. If certain chromosomes occur in the wrong number, if there's too many or too few, the zygote will terminate. It won't be able to survive in order to even grow into a baby. And that can sometimes, well, if it can't grow into a baby, that will result in a miscarriage. Sometimes those miscarriages can happen very early in the pregnancy. Sometimes the sort of the problem doesn't occur until later, and so it could result in a miscarriage later. If there's a couple who are trying to have a child and they have several miscarriages in a row, they'll actually take a sample of the sperm and egg cells and they'll take a look at the chromosomes and they'll take a look at the chromosomes of the zygote and of the fetus after it was aborted to see if they can see any chromosomal disorders. It could be that the mother or the dad improperly makes the sperm or egg cell every time. That would be unusual, but we can actually look for those types of things to try and understand why the couple's having a miscarriage over and over again. So what are some examples? Here's an example. So one chromosome disorder that we can live with and that does result in a viable baby, a child, it's called trisomy 21. Tri meaning 3, the somy talking about chromosomes. So there are 3 number 21 chromosomes in the karyotype. And that results in Down syndrome. In every case of Down syndrome, there are 3 number 21 chromosomes. Another chromosomal disorder is Turner syndrome, and it's where a female only has 1 X chromosome. So what does this mean for an individual with Turner syndrome? You can go look it up. There's another one called Klinefelter syndrome, where you can end up with 2 X-shaped X chromosomes and one Y chromosome, or you can end up with only a Y chromosome and no X chromosome. What does that mean? What does that look like? So there are lots of different um, chromosome disorders that you can live with. Um, a, Another type of mistake that can happen is if part of the chromosome breaks off. So if you think about anaphase where they're pulling the chromosomes apart, sometimes you can actually break off a piece of the chromosome and it gets lost. Um, and if that occurs, you can get different disorders as well. Um, in this example, the cry du chat, you can look it up. It's actually results in a very distinctive cry in French. This is called the cry of the cat. You can actually diagnose this very early, like as soon as the child is born, because the cry is not a normal baby cry. It sounds like a cat. You can look it up. It's a high pitched mewing sort of sound. There are other problems that go along with this, and the life expectancy for children with this syndrome is much shorter than other individuals. But a lot of these we can diagnose um, by looking at the chromosomes before the baby is ever born. So um, I am not going to test or ask questions on non-disjunction, which is what this is called. I just know everybody has questions about, well, what if this process doesn't go perfectly? Because it doesn't always go perfectly. Um, and if it doesn't go perfectly, it results in some sort of chromosomal disorder. Some of them impact life expectancy and quality of life, like with the cry du chat. Some of them have little impact on the quality of life or life expectancy, like the Turner syndrome, and others, like Down syndrome, you can make an argument about quality of life, but the life expectancy is the same typically as other um, individuals without a chromosomal disorder. If the chromosomal disorder is severe enough or it is of a certain kind, the zygote will not ever develop into a viable embryo, and so it won't ever be born. So there you go. That's some information about mistakes that happen in meiosis. And that is the end of our note. Complete the Wiser Me worksheet and let me know if you have questions. I'll see you later.